Topic: blood groups. There are more than 30 antigens on the surface of blood cells. Which blood cells? RBCs. And these will give rise to different blood groups. In a blood transfusion, what we have to keep in mind is the recipient's blood and the donor's blood. It should match. Why? Because otherwise the recipient's immune system will produce antibodies, and these antibodies cause agglutination of the transfused cells and ultimately block the blood capillaries or the blood vessels, which is fatal. When we talk about the types of blood groups, there are two types of blood groups. One is ABO blood group, another one is RH blood group. This is also known as RH factor. RH factor means the factor that is mainly the indication of being A positive, B negative, this RH factor. Taking up ABO blood grouping mechanism first. In this blood grouping mechanism, the blood groups are determined by a gene known as I. I stands for immunoglobulin. It is another isoagglutinogen. Isoagglutino. That means a term for antigen. Clear? So there are three alleles of I: IA, IB, and IO. Protein produced by IA are A antigens, produced by B are B antigens, and O, it does not produce any antigen, so no antigen. Clear? When we are talking about the A, B and no antigens, we discussed that how on a person's blood group these all reflect. People with blood group A have A antigen on RBCs, an antibody B in plasma. Clear? When we have blood group A, it means antigen A and antibody B. Antigen A on RBCs and antibody B in plasma. Let's make a beautiful flow chart to understand that how are these reflected in a human body. Right here we are going to mark the blood groups. It's the blood group. Then it's genotypic allele, that how do these show. G stands for genotype. Then here we are writing the antigen AG. Where are the antigens present? On RBCs. Then I am writing AB. AB stands for antibody, which is in plasma. Then moving to can donate blood to whomsoever they can donate blood, and can receive blood from, where are they act as recipients. Fine. Now if I'm talking about blood group A, it is represented as IA, IO. Just now we mentioned O means no antigen. So it can easily club up with A and give its impression in the blood group. Antigens on RBC corpuscles, it's A. Antibody, it's B. Can give blood to, like donors. Donor for A and AB. Recipient from A and O. We will discuss why. We'll clearly discuss why is it so. Here, while making the flow chart, this genotype will be IB, and IO. Again the same reason, does not carry any antigen, so therefore can reflect a blood group B or antigen B. Antigen is B. Antibody is A. Can give blood to or can donate to B and AB. Can receive from B and O. Now AB. It will be IA and IB. These two are codominant alleles, meaning no one is dominant, rather both are equally dominant. Then blood group reflected is AB. Antibody, no. When both the antigens are present in the blood, then there is no antibody respective to that antigen present. Does not mean that there is no antibody in the blood. Okay, it means antibody respective to this antigen group in the blood is not present. Can donate blood to, only AB, and can receive from all. It's a universal recipient, A, B, AB and O. Fine. Moving to the last one, O. It's reflected by IO and IO, and otherwise also, if not this, small I and small I. Then antigen, none, no antigen. Can we see any antigen here? No. Antibodies, yes, both A and B. And can donate to all, A, B, AB and O. If it has to receive, then it can only receive from O. This one and these two are exceptional cases, right? What is this? Universal recipient, and this one universal donor. This one is a recipient. Clear? Why? What happens? Why this has been made as the recipient and this has been made as the donor? Now, when we start studying these blood groups, we know that every blood group has a specific antigen and antibody in the plasma. Person with the blood group B will have antigen B and antibody A in the plasma. This way we have discussed for all. And whenever this happens, we say blood transfusion made easier following this chart. Imagine we are a storehouse of, or your blood or your body is a storehouse of a particular blood group. We are taking an example, supposingly A. This means if this is a patient's body having blood group A, antigen A on RBCs, then antibodies would be B in the plasma, right? If blood group A enters, your donor is blood group A. Now we are talking about the donors, those who are coming to receive or to donate the blood to this recipient. If it is A, would be easily welcomed because it will be bringing B, and that B antigen, B antibody and B antibody will be easily welcomed. If we talk about blood group B that has to enter, it will be bringing antibody A. Now this antigen and this antibody no doubt are similar to this and this, but are opposite or antagonistic in its function. Antibody's function is to attack every antigen whosoever enters the body. This antibody cannot recognize this antigen as its own, of its own type, so it will automatically attack. What will it do? attack. So, causing the corpuscles to break down, to lies down, which will not be a helpful condition for the body for transfusion. And automatically, this antibody which is entering the plasma of that human being carrying A blood group will start attacking the opposite antigen. So antibodies will start attacking the opposite antigens, and ultimately it would lead to agglutination and RBCs become fatal. In this condition, we have to remain restricted to the type of blood group that is being received by the recipient from the donor. Talking about the third condition where AB comes. AB brings both the antigens A as well as B, but there is no antibody, right? So when it comes as an antigen, as a donor, none of these will be received because these antibodies will automatically attack. So these are only going to receive its own so-called like antibody, not the other antibody or antigen. If O comes, in this condition, when O comes, it brings only the antibodies A and B. There is never ever a fight between the antibodies. Both the antibodies will be welcomed as there is no antigen to be attacked further for. Clear? In this entire summed up notation, we say when a blood group A comes, will be welcomed because this entire recipient body carries the same blood group, that is A, with the antibody B. When a antigen B comes along with an antibody A, this A will automatically break the RBCs or attack the RBCs of opposite antigen. Same way, vice versa for the B. Then if AB blood group comes, it brings both the antigens but no antibody. So the antibody can break or can attack in reverse. And when O comes, no antigen comes, only the antibodies are there, so will be welcomed. According to the given set of instructions, this will be welcomed, this will be rejected, this will be welcome, and this will be rejected. So who two are the recipients for this A and O? Clear? Understood? Now moving to RH. What is this RH? This RH stands for the rhesus monkey, a protein known as, first we write the topic, RH factor. This is the rhesus antigen present on the surface of RBCs in many people. It is discovered in 1914 that this is present in the blood of a rhesus monkey. Hence its name. Depending on the race, 85 to 99% of our population, they have this antigen called RH factor, and for that we call them RH positive. Others who do not have this factor, we call them RH negative. This basically we say American blacks, they are RH positive 95%, and African blacks, they are RH positive about 100%. What happens in this RH factor? It is the incompatibility factor. Incompatibility. When during blood transfusion. Now, we are going to discuss a case of this incompatibility. In this we say, the blood, when transfusion takes place, the first blood transfusion of RH positive to an RH negative blood causes no harm. Why? Because the RH negative person develops anti-RH factor. What it develops? Anti-RH factor or antibodies for this particular one. When the second blood transfusion takes place, ये तो first time था ना, second time, अब क्या होगा? These antibodies will attack the Rh positive factor that has been introduced and will destroy the RBCs of the donor. So during the first time it is not dangerous, but during the second time this incompatibility problem comes, and this is a very important example that we are going to discuss here: incompatibility during pregnancy. Otherwise there is no harm in having Rh positive and Rh negative factor in one's blood group. It's not a deficiency of any kind, but the only incompatibility that comes across is the incompatibility during pregnancy that we are going to discuss here. See, if father's blood is Rh positive, father has a blood group which is Rh positive, and by the chance mother has a blood group Rh negative. Clear? So this is Rh positive and this is Rh negative. If the fetus in the mother's body, okay, that is Rh positive, then the problem occurs. This is a serious problem. Why? Because the Rh negative blood of the mother is not earlier come in contact with the Rh positive blood of the fetus during transfusion. So her first child will suffer no problem. Why? Because Rh positive blood of the fetus stimulates the formation of antibodies or anti-factor in the mother's blood and try to destroy, but at that moment of time the anti-Rh factor or antibodies are not yet enough that they are produced to harm the fetus. But whenever there is subsequent Rh fetus there in the mother's body or during successive pregnancies, the anti-Rh factor or antibodies of the mother's blood can destroy the RBCs. This results in hemolytic disease of the newborn, that is known as erythroblastosis fetalis, destruction of erythrocytes. The newborns may survive, but with severe jaundice, and generally what happens is, in order to prevent this kind of problem, Rh negative mothers are injected with a defective anti-Rh antibody during all pregnancies to carry Rh positive fetus. So Rh positive, when it becomes dominant, it creates problem. The first child will remain normal, but during the second child, mother will develop a kind of anti-Rh positive factor, that means the antibodies to kill this factor, in her blood. Why so? Because after the first delivery, some of the Rh positive cells are exposed to the mother's blood through the placental cord. And what will happen? Mother is not at all exposed to this blood earlier, so she did not know about this kind of antigenic problem. She will start preparing antibodies. Which kind of antibodies? Anti-Rh antibodies to kill these positive antigens which are there in the fetus. Ultimately, what will happen? During the second pregnancy, when everything is going normally and there is transfusion between the mother and the fetus blood, these Rh antibodies will also travel. And if these anti-Rh antibodies are dot-like structures, they will move inside and will kill the fetus RBCs, which leads to erythroblastosis fetalis, leads to severe jaundice and anemia. This way, the second pregnancy proves fatal for the mother, not the first one. Alright, this is known as incompatibility factor, where the female has to be administered later on with defective anti-RH antibodies to protect her fetus from this ongoing problem. So in this case, when there is an anti-RH mother and RH positive, this RH negative mother and RH positive fetus, then first pregnancy remains normal but the second pregnancy shows a fatal result if the antibodies are not treated, if the mother's blood is not treated prior to that. Clear?